Hello and welcome to Histone Modification 101, the crash course that'll teach you all about histone modifications. Histone modifications are one of the primary methods known in the field of epigenetics, which deals with understanding the factors outside the DNA that influence gene expression. Let's begin with the basics. Deoxyribonucleic acid DNA contains all the necessary instructions that are needed by organisms to produce proteins, but there's a lot of it, so what happens? Due to the sheer amount of it, DNA is packaged by histones to form chromatin, which is then further condensed to form chromosomes. These histones are then strung together to form a complex called the nucleosome. The histones can undergo modification that can determine whether gene expression of a certain gene is on or off, which impacts the way genes are expressed in different people. The chemical compounds that modify the genome are called writers and erasers, and are very important to the system. The most researched of these modifications are acetylation, methylation, and phosphorylations. Others include deamination, ADP ribosylation, ubiquitinylation, sumylation, and histone tail clipping. Acetylation, which is done through HATs and HDACs, works through opening the chromatin to allow transcription to occur. Similarly, deacetylation allows for a closed chromatin state and results in transcriptional repression. Methylation is very diverse. Arginine methylation promotes transcriptional activities, and lysine methylation both promotes and represses activities. It's carried out through HMTs, PRMTs, and KDM. Phosphorylation occurs with the help of protein kinase writers and phosphatase erasers and is thought to facilitate gene expression by reducing histone affinity for DNA, which allows for better transcription. There are two main modes of action looking at histone modifications. The first involves directly influencing the structure of the chromatin at varying distances, and the second involves positively or negatively regulating the binding of effector molecules. Wondering if these modifications can occur in any genomic region? If so, here's your answer. There are two genomic regions. Euchromatin is known as the open region, which includes most of the active genes that undergo modification. In contrast, heterochromatin regions are relatively compact structures that contain mostly inactive genes. Because of the large number of possible modifications, there's a regulation system in place that keeps everything running smoothly. In addition to this, however, is crosstalk, which allows for fine-tuned control of modification and exists through multiple mechanisms. Modifications play a role in many aspects of a person's life, such as diet, atopy, and inheritance. In modern-day studies, understanding what each modification does allows for the possibility to create new epigenome-based treatments. The possibilities are truly limitless.